All right, everyone. So I think it's time to start again with our afternoon talks. And so the first afternoon speaker is Doug Yu. So Doug is a professor at the School of Biological Sciences at the University of East Anglia, and he will be discussing the use of eDNA when managing wildlife with data on salmon, leeches, insects, and forests as case studies. So Doug, whenever you're ready to share your screen.
How's that?
Yeah, that looks great.
Okay,
And
so I just slight change of title. Thank you very much, Eleni. You can hear me fine on the screen?
Yeah, I can hear you. I did this for all of the speakers, just trying to make it seem like I'm not just reading off someone's slide. All right, so whenever you're ready, Doug.
Okay, all right, thanks very much. Today I'm going to talk about a few different studies we've been doing over the last few years. And what draws them all together is that they are all high dimensional data sets. So lots of columns, lots of rows. And the point of these high dimensional data sets is that they give us more power to answer questions that we have been wanting to answer. So let's start with the simplest example, which is in Alaska. So that's Alaska there in the red. And Alaska hosts a salmon fishery that is worth about $500 million a year. And the salmon are captured in the ocean. And so then if they escape the capture, they get to go up to the freshwater stream, they change their bodies, become reproductive, and then they breed. And so that breeding population is known as the escapement. And the real question for management is whether or not the fishery is allowing enough of these fish, the salmon, to escape. And so you have to somehow figure this out. And what the Alaska State Department of Fish and Game do is they actually count these fish. And so they have a model about what they think is a reasonable number of fish, and then they track it over the weeks. And then every Friday or so, I think it is, they issue closure notices and opening notices for different stream openings so that they can close the fishery for years, so to let more fish go up, and then they open other fisheries and so on. So this is a closely managed, resolved fishery. But there's a problem, which is that there are, just in this part of, known as the southeast of Alaska, it's where the capital is, there are about 1,000 streams that are used for breeding, as estimated. And they have a counting station on 14 of them, right? So that's 1.4% of the streams. And the reason for this is pretty obvious. Most of what you're seeing there, even though the capital is there, most of it is you can't get to without taking a boat. There are bears. It is not an easy place to work. So they're really managing a huge fishery based on a tiny amount of data. And so we thought, well, one of the reasons that it's difficult is because it takes a lot of effort to count fish, right? So here's the best counting station, which is in the capital. And you see multiple people who are there all day long, and catching the fish, sexing them, weighing them, and so on. So they do more than just the simple measurements, and then they let them go upstream after this. And so they do this all summer long. And we thought, well, is it possible, right, to get the same kind of information, at least the count information, with just one sample per day using water? Of course, fish release DNA into the water, and then you can count the copies of a particular sequence that is specific to that species using quantitative PCR. And so you need count data, which we get from the Ock Creek Research Station. And then we get eDNA, and it's terrible, right? If you look at it. Okay, it's not completely terrible. When there's no fish, right, there's no DNA. When there is fish, there's DNA, but they don't really match each other. So what's going on here? What's going on is that eDNA, these measures by quantitative PCR are concentrations. So it's some kind of nanograms per microliter type thing. But the thing is, the number of microliters is changing because the level of the river is going up and down all the time, as you can see here. And so what you really need to do is you really need to multiply the volume of water times the concentration to get DNA. So it's America, so they measure water per second, so it's really flux, as cubic feet per second. So if you do that and then you do a little rearranging, you get nanograms of DNA for that species per second. And now actually it looks pretty good, right? So when there are a few fish, there's low amounts of DNA. total volume, and when there's lots of fish, there's lots of DNA. And then you can just fit a model and ask how well does this DNA rate explain the variation it counts, and you find that the model is highly significant. You can just see by eye that it is. It's not perfect, and I can tell you right now that the primary source of error here is not the DNA, it's the water volume measurement. That's much harder to measure, actually. And then the other thing we did was we tested, because we have data from 2015, 2016, it's only useful if the model's parameters remains the same, right? So we only have 2 years. We did an interaction test by year, and we find that there's no significant difference in the interaction term, no significant interaction term. So the parameter values between the 2 years are not significantly different from each other. So that gives us some confidence that if you were to parameterize this for a few years, you'd have a model that works for that river. And then potentially, if you did this for many rivers, maybe added some extra covariates, you could create a robust model for many streams. And then it becomes possible to contemplate setting up machines to collect water, or citizen science networks to collect water, and then be able to track the fish over time. So the eDNA does contain really precise abundance information, and because it's easier to collect water than it is to count fish all day, it's potentially possible to manage this much more closely. Okay, so that's a pretty simple example using a pretty simple technique, qPCR. But I'm a more terrestrial ecologist type person, and one of the questions that's always animated me is how we know whether a nature reserve is effective. And so what we're really asking is whether or not a nature reserve contains the animal that it's supposed to contain, right? And as you know, this year, China and Vietnam have announced bans on wildlife, and the question is, will this be enforced? Will it have an effect? Will it have an upstream effect on wildlife in nature reserves? So it's really difficult to measure wildlife, by which I mean vertebrate animals. And this picture is from the Vietnam rainforest, and there are 2 large mammals in this photo. And I don't know if you can see them, but there's one right in the center, and it's a human. It's one of your own team as you're walking up a stream. And you can imagine that if it's so difficult to see humans, it's really difficult to see animals that are trying to stay away from you. Never mind different types, right? So you have frogs and reptiles and mammals and birds and so on. So we thought, well, why don't we try doing something where we could try to survey wildlife efficiently using DNA? And we chose a place called Ailashan Nature Reserve, which is basically the mountain ridge in China, and it's about 700 square kilometers. So it's almost the size of Singapore. And we want to know what animals are present and where are they? How are they distributed in this nature reserve? And so the way we decided to collect the DNA was by collecting the collectors. So in this case, we collected leeches, and basically the leeches are trying to collect you, right? And then they carry a little bit of blood from their last blood meal, trace amounts. So that's the eDNA. And then you go take them into the lab. We got 30,000 of these leeches collected by 160 forest rangers over about 3 months. We choked on that for a while and then sequenced them. And so here they are. And then we issued the rangers with these little hip packs. And then each hip pack had bags in them, little Ziploc bags in them. And so it naturally induced the rangers to put leeches in one tube, and then put leeches in the next tube, and the next tube, and then the next bag, and the next bag. So we had repeated samples, and then you see these little areas, and that's one. These are 172 patrol areas. So we asked rangers to collect leeches. So we had multiple samples from multiple patrol areas, and then we took those leeches and we amplified two different genes, 16S and 12S. 16S is more devoted to mammals, trying to pick up more mammals, and then 12S is a sort of general vertebrate one, so we're picking up amphibians and so on. But actually, both of them pick up quite a bit of everything. The 12S is better at picking up birds. So to sum that up, right, you issue these bags and tubes with RNA later to park rangers who 30,000 leeches in 3 months. There's a whole bunch of molecular lab work, which I'm not going to go into, in bioinformatics. The bioinformatic pipeline itself is the subject of like 2 or 3 papers. And I just wanted to point out that we use something called PROTACs, which is a software, statistical wrapper that helps you assign taxonomies and identify species that are not in your reference database, and say this is a not present in the reference database. So you have to give it a SPA in its name, and then took that huge data table of which species we detected in which replicate in which patrol area, and applied something called a multi-species occupancy model. And so a multi-species occupancy model tries to separate and model two different things, right? A species may or may not be present in a place, and even if it is present, it may or may not be detected by your leeches. So that's essentially two processes, right? So we have a coin flip where the coins, the chances of being present or not present is dependent on some environmental conditions, right? So in our case, it was elevation and distance from reserve edge is the environmental conditions, and then that tunes the coin flip. And then if it is present or not, then do you detect it or not? And then what tunes that is, in our case, we used sampling effort, so the number of leeches in your tube. So you don't have to try to record like how hard or how well the patrol, the ranger was looking for animals. You just count the number of leeches that are in the tube. And then there's some hyperparameters, which I won't get into, which are shared across all the species. That makes it a community model. So we ended up getting leeches from across the reserve, not in every single place, but across the reserve. And then you can apply the multi-species occupancy model, and you can model the estimated species richness across the entire reserve based on its environmental covariance, elevation, distance from edge. And as you can see here by eye, you should see that there's more, that we detect more species of vertebrates towards the center of the park, right, towards the core, and more towards the lower half. That's the higher elevation part of the park. And you can see this if we take each point, right, with its estimated species richness, and it goes up very high. And if you can also model whether or not the species richness goes up with distance to edge. It does less, more noisily, and that's because these things are correlated, and elevation does most of the work here. And then you can do more than just the estimated number of species. You can actually measure or estimate the occupancy. So the occupancy is the probability that a species is in one of these patrol areas. And as you can see, the probability, the average over all the species, goes up with elevation. I've separated them. So here you have the reds, which is birds and mammals, and so birds and mammals in blue, and then amphibians and reptiles in red. And amphibians and reptiles generally have higher occupancy. They're more spread out, more available everywhere, which makes sense because there's more frogs, basically. And then the birds and mammals also show that. So they're going up. Now, if you look on the right-hand side, you also see that preponderance of lines going up, which is that each one of those lines is a separate species. But you definitely see some lines going down as well, right? So most species are found more towards higher elevations. Some species are definitely found towards lower elevations. I'm going to do the same analysis for the distance reserve edge, and it's much weaker, or maybe not present at all. And this is because because the distance to edge and elevation are correlated with each other. Okay, so we don't want to know just the whole community. We want to know each individual species and how they are. And so muntjac niginalis, which is the northern red muntjac, is known to be resilient to hunting and so on. And it is mostly found in the lower elevations near the edge of the park, and there's more open area, which is what deer like, of course. And then here's another example of a leopard cat, and we find its modeled occupancy is higher in the center of the park, and despite the fact that it eats rodents. You can do this for all the species. So this is all the mammals in the data set, right? And if the mean is above 0, which is most of these points here, that means that its occupancy goes up with elevation, right? And if the mean is below 0, that's just a few on the left, its occupancy goes down with elevation. They like low elevation. And so we can ask, what are those species that prefer to be at low elevation? Well, 3 of them out of the 5 are domestic animals. And these are species that are taken in by neighboring populations of humans and allowed to range in there. And so you can see them yourself. This is a camera trap photo. Northern red muntjac, the field rat, right? And these are species that are resilient or to hunting or not hunted at all. And we know that there is some hunting in this park. So it makes a lot of sense that the rest of the wildlife is arranged towards the center of the park. So to sum up, and I'm not showing you most of the results here, most of the birds, most of the large and small mammals, right, except for these exceptions on the left, are found more in the high elevation sites. And and we found that was also true, of birds. We did not find that was true for frogs, which kind of makes sense. So the leech-derived eDNA or iDNA lets us survey an entire reserve in 3 months. It's taken us a few years to work out the whole pipeline, but now we have the pipeline, so we can repeat this. In fact, we are repeating this with another set of leeches. And the only thing stopping us now is that every sequencer in China is busy sequencing COVID genes or something. And high throughput, you can do all the different kinds of taxa and the vertebrates. And if you wanted to, we can actually go through these same leeches and go look for viruses as well. We know we can do that as well. So, but going back to the original question, the nature reserve does preserve biodiversity. We see that it's working, right? But there's clearly a lot of room for improvement. And we know now where that improvement should be. Okay, so just want to thank Yingqiu Ji, who worked the molecular pipeline, most of the bioinformatics. I did some of the bioinformatics, and then Chris Baker, who did the multi-species occupancy model. And then Wenqing Zhong is the vice director of the Yunnan Institute of Forestry, who's in charge of these parks. And he came up to me after a talk one time and says, you can do this stuff with leeches. Why don't you do it in this park with us? So this is actually all his idea. All right, so if there are leeches, you can do this kind of thing in a park in China, but can you do this in a much more difficult place, right? So like the South Midlands. So this is the same area that Naomi Ewald was showing earlier. I think probably the same set of samples, actually. And there are many, many fewer blood-sucking leeches in Oxfordshire because they're in Parliament, obviously. And so there's a lot of semi-natural habitat here, right? And but it's a really difficult job trying to sample this. If you wanted to know where all the vertebrates are, you can't just go around sampling. You can't put up cameras and so on. And there's no leeches. So we took advantage of this private industry sampling, pond eDNA for great crested newt. And that's just to go over what you've seen before, right? You build a house near a pond, you have to sample that. And nowadays what you do, of course, is you contract some company to go and sample the water, and then they take that DNA and they look for qPCR, using qPCR, look for great crested newt. And then on top of that, an organization called Nature Space Partnership brought together NGOs and the government and so on, and systematically sampled 600 ponds across the South Midlands. And then they used, this is using qPCR on eDNA, and then they built a single species distribution model across this whole region, right? And then turned that into 4 zones, white, green, red, and amber. And so you pay a certain amount of money depending on the occupancy of the great crested newt. So there's lots of DNA samples sitting in freezers. And we asked Naturemetrics if we could have those samples. And out of the 600 or so, 305 still had some DNA. And so we were able to go and just take the samples, metabarcoded them. So I figure it's at least 150,000 pounds worth of sampling. Thank you very much. And my student, Tai Wong, found these species. So we got domestic animals, birds, fish, amphibians. So again, metabarcoding, we get a whole range of vertebrates from one single sample, including the great crested newt. Now, we want to now understand what's determining the distributions of all these species. And there are lots of different ways of doing that, but we're using something called joint species distribution modeling. And because it's a fairly large data set, we use a FAST method, which is not yet published, but is available, called SJSDM, which is an R package. It uses machine learning in the background and GPUs, and I've burned a lot of calories doing this. But basically, we have a lot of local environmental variables taken at each pond, but because that's what you have to do. And then we also have land cover around 500 meters around your pond, to sort of what's the dominant land cover around each pond, right? And then the spatial location. And then, so I'm going to just show you some of the results. For the 5 amphibians, SJSDM is telling tend to be found more often in smaller ponds. The other 3 amphibians don't show a preference. Smooth newt and gray crested newt tend to be found more often in ponds that have high macrophyte cover. The other 3 don't really show a preference. But when you look at the land cover, common toad frog and palmate newt tend to be found more often in ponds that have woodland as the dominant habitat type around them. And that makes sense for palmate newt, especially because it's sort of a more northern species. And maybe this is what keeps it cooler in this area. And it turns out this lovely thing about JSDM is it's doing this for all the species at times all their various environmental covariates. And the SJSDM doesn't really worry about collinearity, so you can just pile in all of the different environmental covariates, and it's not a problem. So we have a niche description for all the different species that we found in the thing. Now, you can go over those one by one, but you can also look at it a little bit higher up and ask, what is governing these species overall? And we can look at something that's been called the internal structure of metacommunities. And so this is a ternary diagram. And what this is doing is for every one of these species, that's a point here, right? Is its distribution better explained by environmental covariates, in which case it would be like these species here, which are mostly fish, and the environmental covariates that we have in hand, right, do a really good job of explaining the distribution. Here's a bunch of species that are poorly explained by the environmental covariates that we have, but they tend to be found in ponds near each other. So they seem to be dispersing back and forth, but the environmental conditions might be very different in these 2 nearby ponds. That's spatial autocorrelation. And these are birds and mammals, so it makes sense because they live between the ponds. They're not pond dwellers. And then there are a few species where not autocorrelation, it's not environmental covariates, it's something else that we haven't measured or some interaction between the two. And, but in general, we're finding that most of the species are down towards the left, towards the filtering. So that's suggesting that we're doing a fairly okay job of explaining the niche outlines of these species. And the better that we're able to do that, the better that we're able to explain the niche requirements of the species, we're better able to do things like that mapping, but for multiple species at once. And that could be then used as a multi-species offsetting, multi-species restoration, priority setting, that kind of thing. And so it looks promising. So a couple meta lessons, right? Industry is paying large amounts of money to sample the UK landscape. Naomi was saying that you're moving to metabarcoding. I am absolutely in favor of this. And also said that you're using filters to sample water rather than the ethanol, and absolutely in favor of this. I think we captured fewer species than we could have because there was just less DNA in every sample if you use ethanol. So use filters. Please, Dr. England, allow filters to be used on great crested newt samples. And also, I really think it's important to take 2 or 3 samples, separate samples per pond to allow occupancy correction in the same way that we did for the leeches. Okay, 4th, last case study. So this is more pure science, but it's a very basic question: how will biodiversity respond to climate change? So this is somewhat related to what Francisco was saying, but he was working backwards in a way, reconstructing. We're trying to see, can you work forwards, right? And the standard way of trying to predict how species will respond is something called the correlative approach, which is that you have a current climate with climate envelopes, you have a future climate, right? And then you have a species distribution now, and then you move its distribution in line with its climate envelope. So this correlative approach is the most frequently used because that's the kind of data that we have. Everybody knows that it is incomplete, right? Everybody knows this is not a controversy. It's just that how do you bring in something like interspecific interactions? Where do you get the data to do that kind of extra model? And so it's really, really tough. And so here's a potential way to do it. Luckily, a few years ago, I was brought in to work with a group of Scandinavian biologists, and they have been collecting insects in Zackenberg Research Station up in Greenland for the last 20 years or so. And it's mostly a spider, fly, wasp ecosystem, as you can imagine. There's not a lot of vertebrates up there. And they collected these insects and spiders in pan pitfall traps and then systematically have been sorting them and keeping them in a warehouse in Denmark and in alcohol. Right, and so they've only ever been identified to family level. And there's like 750,000 at the time we started to do this project of these little bugs in tubes of alcohol, right? And we know basically what the species richness is. So we have samples of them. We managed to get 308 mitochondrial genomes out of the 375 or so species that are there. And then we took every trap week from 1997 to 2013. That's 492 trap weeks. And then shotgun sequencing. That just sequenced all the DNA in that sample. So that's reconstituting a lot of samples. And then we did another 200 or so technical replicates. So that's a lot of sequencing. Just downloading the data from the sequencing center took days. And that's on high pipes. So we mapped the Illumina reads. You took the sample, you took all the reads, and you can just map them to each of the miter genomes. So if this species here, that's this miter genome on the top, and it attracts a lot of reads. So we can say that species is present there and high biomass, right? And this species is also attracted reads, but it's only attracting reads to a small portion of the miter genome. So that's some conserved sequence that's probably common to other species. And so we would say that that species is not there. That's a false positive. It's very easy to separate false positives from true positives. And we've done tests, and it's like we can do a perfect separation. And so when you do all this, you spend a lot of time on the computer, what you end up getting is what I'm calling a community time series. So you're going from left to right. Here's just a portion of that data set, 1997 and 2013. Going from left to right, you'll see pink, white, and red boxes. So red means a lot of biomass of that species in that timeline. And I'm only showing the first and last years. The other thing you should see is that summer is twice as long in 2013. That's global heating. And so we're able to not only see presence absence, we're able to see, because we have all these various controls and so on, changes in phenology. Okay, you can do some summaries, right? And temperature has increased by 2 degrees since the beginning of their time series. And what was surprising to me is that species richness has declined by like 50% during this time as well, which is, I did not expect this. And so we'd like to know why, right? What is governing all this change? And if we go back into that data set, the correlative approach, that simple sort of move the climate envelope approach, would do something like this. Here's a species, that blue square, and it's got a biomass peak, an activity peak in early August. And if you go to 2013 and go to early August, it's not got a biomass peak anymore. But as you can see, there's a biomass peak kind of, in July. So that makes sense, right? There've been more degree days, it's been warmer. So this species of chironomid will emerge earlier and show some more activity earlier in the year. So that's what you would expect if you're just moving climate envelopes. But it's not actually that easy to find something like that. So here are 3 more species active in August and nothing, right? They're not active in August, they're not active earlier. Like there's a whole bunch of species that showed an activity peak some point in 1997, and then you look in 2013 and there isn't one, right? So they just disappeared or became very, very rare. And there's a whole bunch of species that didn't show activity peaks in 1997 and do show them in 2013, so they became abundant. So this is not so obviously easily explained by just temperature changing, right? So what's going on? We're lucky, I said, because not only do they have this great data set, but we also have the people who literally wrote the book on joint species distribution modeling, Nerea Abrego and Otto Vaskainen. And so we have this paper in review now where they modeled the presence or absence of each species every single week in a year, all right, as a function of temperature of the previous month, temperature in that week. We've added recently the week number to capture the phenology, week number squared to capture the fact that it goes up and down. And then also last year's total abundance of that species. So is there some intraspecific density dependence? Plus last year's abundance of all the herbivores in our data set summed together. Plus last year's all the parasitoids in our data set summed together. plus last year's all the predators, that would be spiders, for example, summed together, and then detritivores summed together. And we've done this on a pairwise basis as well, but this is an easier version of the model to look at. And so these last 5 terms are potentially species interactions from last year. And you can do this because we have a community data set. So we're tracking multiple species at once. And then, of course, you're doing this jointly for multiple species. So they're sharing some error terms, what makes it a joint species distribution model. And so that's a lot of terms and a lot of cells to fill. And so then you can ask, do these species interactions play any useful role in predicting presence absence from week to week to week, right? And so what you do is you compare 2 models, the one with just the abiotic terms, so that's the correlative approach, right? And the ones with species interactions, right? So that's the full model. And then you can compare in 2 ways. You can say which model fits better. That's the explanatory power. And just all you need to know here is that explanatory power is measured from -1 to +1. If it has a positive number in the power, then the model does better than average or better than random, right? If it's a negative number, then it does worse than random. So the abiotic model does a better than random job for all but 4 species. It does a worse than random job, which is kind of amazing, for 4 of the species. The interactions model, the big model, does better than random for all the species, and in general does better, right? It's got a 25% improvement in explanatory power. Okay, but you could argue that the more complex model has more terms, right? So here's another more stringent test. You can fit the model to 13 of the years and ask if that fitted model can predict the presence absence from week to week for the 1 year that you held out. And they're doing this for all the different years. Okay, and so that's predictive power that you're measuring. And in general, prediction is harder than explanation, right? So the abiotic model and the interactions model do worse. So there's a lot of dots that are in the negative zone, but the abiotic model does terrible, right? It's better than random for 20% of the species, which means it's random or worse, right? In other words, it's predicting presence when it's actually absent, or vice versa for 80% of the species. The interactions model is better, relatively speaking. It's better than random for 50% of the species. So it seems really quite useful to have other species in your model. This is evidence that species interactions are helping to govern presence and absence as climate has changed, right? And the correlative approach, which is the abiotic model, won't tell you that. So I'd say, the conclusion here, right, is that the trophic interactions model has better power, but it also means that the correlative approach generally in predicting species responses to biodiversity responses to climate change has got real problems with it. So I don't have a great answer to like how biodiversity generally will respond to climate change, but given that it's possible to create these community time series, more and more easy to do this, right? We should be able to discern more and more the effects of species interactions. And it's taken a lot of effort, right? I mean, the vision of being able to collect all that stuff started in 1996, right? Through to be able to solve the bioinformatic and molecular problems to the statistical analysis. Okay, so to sum up, 4 case studies. And I think what's going on here in ecology is that we're starting to see a convergence of technology, remote sensing, new kinds of data, right? Creating these high dimensional data sets that are allowing us to produce these more complex and powerful models. But I think the other lesson is that ecology is transitioning, right, away from being a basic science into something more like an engineering discipline, right? So we're not studying movement, we're studying automobile in manufacturing. And when you try to do that, the quality control becomes more important, instrumentation, use of instruments in general becomes more important, standardization of protocols, I think is really obvious, automation as much as possible, right? And most important of all, scaling up, being able to scale up in terms of production of information and results. So I think that's where we're going. Okay, thank you very much.
Thank you, Doug. Of course, super, super interesting. So you've taken it to another level with the leeches, I think. You're not relying on direct observation of the species, but even you're looking at leeches, and then you're going forward in the future to understand the effects of climate change. So it really highlights how widely applicable these methods are. We've got a couple of questions in the chat, and I think they both relate to detectability. So one is for your leeches data sets. How do you account for differences between how likely species are to be predated by the leeches?
Right. So you could try to come up with a per species detectability thing, and but then you'd have a lot of parameters to fit. So in the balance of that, we ended up separating whether a species was a cold-blooded or warm-blooded, and then fit to those two groups. And if we are getting more data, and so hopefully over time we'll be able to fit more complex models. But these are sit-and-wait predators, right? So they seem to be, they eat whatever is around, whatever they get onto. So that's the best I can give you right now. And then the second question.
Yeah, so it's again about detectability for especially JSDMs. So Chris says detectability is a real issue with eDNA. So that's why you used occupancy for the data set in China. But you showed how imperfect detection JSDMs detections seem to be perfect. Is this an issue for how confident we can be in the results based on the assumption of perfect detection?
Well, I mean, I'm not the person to answer this, actually, and Jim Griffin, yeah, is the holy grail in the statistics is to merge occupancy modeling approaches with joint species distribution approaches, and so they are doing this right now as we speak. In fact, the person who is doing this isn't even watching today's talk. He's busy sending me questions on Slack on how to.
I noticed.
Data sent to him. So yeah, so that's your answer. Alex Diana will tell you in a few months.
Yeah, so that's a point to make. Maybe Jim will mention it as well, that we have a NERC project looking at exactly that. So Jim will describe the single species case, but the multi-species case is quite challenging, as Doug's saying. If you want to account for observation error, how do you want to do it per species? What if you have hundreds of species? So that requires a little bit of thought. There's one more question from Malcolm. In reality, it must be chaos in the PCR during amplification. All these quantifications assume independent responses to primers and PCR components. How confident are you that your relative quantifications are not, to some extent, a consequence of different compositions of species, resulting in different interactions of primers to specific species templates?
Okay, so there are two different kinds of data sets we have here. The leech one is using PCR, and there is potential for some competition amongst things. But the best person modeling PCRs and so on, Amy Willis in the University of Washington, is found that the data fit better with the idea that there is no interaction during PCR, that there is just some unknown species bias, species-specific bias for every species, which is estimable. In fact, they have software to estimate that. And so, what that means is that you actually have to break abundance apart into two things. So we're used to counting something and then be able to say this species is more abundant than that. But the other component of abundance is that we can say within a species, it's less abundant, more abundant, less abundant, more abundant, right? And you can say that, but you can't really, so you can say slopes, but you can't really say intercepts. You can say, it's hard to say that this species is more abundant than that or vice versa because of all these hidden biases. But the bias is the same for within every species, so you can do this. The second data set was that insect data set from Greenland, and that's what we took advantage of. So we're mapping, we're not doing PCR, there's no PCR in that situation, but there's still biases. And we made mock samples, and we were able to show that we were able to reconstruct within species of differences in abundance from mock samples, right? Like almost perfectly, once we do a bunch of corrections and controls and like that. So that's most of the abundance information that you need, actually. And Nerea and Otso did that same model they showed you using presence absence, and they also did it using DNA abundance. And actually, presence absence works better in that particular model. But abundance is one of the things I've been obsessed with. Yeah.
Okay, so my headphones are dying on me, but maybe I've got one more. We've got time for one more question. So, Chibuzoro, sorry if I killed your name there. Thanks a lot for the brilliant presentation. The leachy DNA detection approach is quite innovative. My question is on the occupancy modeling. In your two-level occupancy modeling, what other factors could you have control for apart from elevation? Might worth checking how the results will change after adjusting for some other covariates that vary with elevation, example, temperature.
Okay, so we had a few other covariates to use. There are some roads going through, some streams, and so on. And maybe the streams would have been better for doing the amphibians. Before fitting the model, there was a process of Bayesian model selection. And the Bayesian model selection suggested that for one, we have two genes. So I only showed you for one gene, but one of the genes was distance from reserve edge, and elevation would be the only covariates worth putting in the model. And the other gene, elevation, was the only one. Elevation is a summary variable for lots and lots of different things, right? Temperature, minimum temperature, high temperature, aspect, all sorts of habitat variables. So elevation itself, it explains a lot, but that's because it's telling you a lot, right? It just so happens that in this park, elevation also tells you, position in the park as well, right? Because of the way that park works. So I'm happy with the fact that it went simple. The way to go further with that is to get a separate data set for particular animals, maybe a camera trap transects, things like that. That would be the best way to check.
Okay, Markham says thanks. He's now reassured. So I'm
glad.
Was it your hand in the video with the leech on it moving around?
No. I don't let them bite me. They don't carry diseases, which is nice to know. But you do lose a lot of socks to the washing machine.
Oh god, it was pretty gross. Okay, so I hope I haven't missed any questions in the chat. If I have, please. Oh, there's another message. No, just thank you. Right, so if I've missed your question, then just maybe use @ so to direct it to Doug, and maybe he can reply to you afterwards. Thank you very much.
Or you can send me a question on Twitter. I'm an insect soup. So.
Oh, yeah. Super. Thank you very much. It was a amazing talk, collection of case studies. So I think now it is time, isn't it, for the next talk? Yeah, so 1:30. So the next speaker is Jim Griffin. Jim has been teaching all morning, but he's very kindly still agreed to give this talk this afternoon. So Jim's a professor of statistics at UCL, and he'll be presenting the model that we've referred to a few times this morning. So it's a Bayesian model for eDNA data that accounts for false positive and false negative errors while performing variable selection.
Thanks, Eleni, for organising the meeting. Yeah, as Eleni was saying, unfortunately, I couldn't make the morning session, so that was a real pity. Yeah, so to kind of get into things, so as Doug kind of previewed in a way, we've been working on errors in eDNA data. So the initial work was looking at single species models, and so we did that work with Eleni and Richard Griffiths and Andrew Buxton at Kent. And so now we're moving into multi-species models with metabarcoding, and we're working with Alex Diana and Alex Bush and Doug as well. Okay, so what I'll do is I'll talk mostly about the single species case, and then I'll say a little bit about the multi-species case and the extension, and then kind of conclude. Okay, so I think a lot of this probably people have gone through today. the importance of monitoring wildlife species, and some of the challenges that are faced with that. So in particular, as Doug was saying, we are not sure whether there's, well, we observe species at particular sites, but it may be that the species is actually there, but we don't observe it. So that can be particularly challenging for particular types of species. Also, in terms of monitoring, in terms of people doing kind of physical monitoring and counting of animals or observing of animals, then that can be very challenging, time-consuming, expensive. And to scale things up to large numbers of species, we would need to have some kind of remote sensing. So eDNA is one example of that, and one example of trying really to move beyond sampling methods into technology-based approaches. Okay, so I guess for sure people have been through this. Environmental DNA involves taking a species and in some way trying to measure the DNA in the species. Okay, and so it's becoming increasingly used for a wide range of different problems. But one of the issues with eDNA and a lot of remote sensed data more generally is a question around data quality and how reliable that data is. And as a statistician, kind of an interesting part of that is how we can try and measure where these errors are coming from, what causes them, how large they are, and also how we can adjust inference for these kinds of errors. So unlike in a lot of situations, for example, in occupancy type models, in eDNA, we have the possibility of both false positive and false negative errors. So in the usual kind of occupancy models, people would assume false negative errors. But here, people would like to assume both types of error. And in a way, there's two kinds of ways that errors can arise, well, at least. So one is in the data collection stage, and the other is in the analysis stage. So we can have errors in both stages, and potentially we can have more stages. So if there's a kind of complicated process, if the analysis involves a complicated kind of workflow, then it may be errors are introduced in all sorts of different stages of the analysis. And so depending on the size of those errors, if they're not small, it would be useful to take them into account in our inference so that we can adjust for any kind of biases, or we can include it in the uncertainty in our estimation. Okay, so what I'm mostly going to talk about is the work that we've done on single species. So in terms of single species, really I became involved with this through talking to Eleni about problems that people were having in Kent with qPCR data and eDNA samples. So that's kind of the background of where I kind of came into it. Okay, so what people have is they have measurements of qPCR. And so usually what people do is to take multiple runs of qPCR for different samples to try and understand whether there's presence at a site. So this is a kind of an example of some data. So we have different ponds. So here we're interested in great crested newts ponds, and we have a score, this eDNA score, and that tells us out of how many replicates, PCR replicates, how many positive results do we get. And so you can see that we get lots of things that are either 0 or 12. But for example, for the observation in the middle, the third pond, we have a measure of 7 out of 12. So there we've got something kind of strange going on, that we can't really be confident of whether there's eDNA there or not. really. And in a way, people often will try and take these eDNA scores and then turn them into occupancy problems. But it seems sensible to try and at least take into account this uncertainty here. And that will allow us to understand the quality of the methods that we're using. Okay, so our kind of contribution into this area was to build a Bayesian model for single species eDNA data that can account for both false positive and false negative errors in both the data collection stage and the experimental stage. So there's already been some work in this area, and there's been work on multi-scale occupancy models. So we kind of extended that work and addressed or came up with a way to address some of the issues around those models. So I'll kind of come on to what I mean by that. On top of the type of data that we had in terms of eDNA, we also had, for some of the sites, we had confirmed species presence. So somehow through records of physical presence, certain sites, it was known that species existed. And so we'd also like to take that into account, okay? So this is the model that we came up with. Well, it's really the multi-occupancy species model. So that idea is that if we think about a specific site or a specific pond S, then our observations, so we observe, what I have here is KS, so we observe sometimes confirmed presences for some sites, and we like to take that into account. And also we observe YSM, and that's the number of positive runs, PCR replicate runs from K possible ones. So something's missing from my slide here when I edited there, is that on top of sites, in the model, we assume that you may have multiple replicates from a particular site. So not PCR type replicates, but that you would collect different samples at the same site, either by going at the same time and collecting samples in different places, or by visiting multiple times to collect different samples. So that's what M is referring to, these different sampling occasions. So I call that a sampling occasion. It's not technically a sampling occasion, but a different sampling occasions. So we've got two kind of sources of information. Our main source is from the eDNA data. That's these positive PCR runs, which is the YSMs. And we have confirmed presences, which are the KSs. And then the things that we would like to infer. So one thing is ZS, which is the species presence. And also we assume this WSM, which is that eDNA is present in the nth sample. So it may be that different samples, if you take different samples in different places or in different times, that you're going to have different records of eDNA being present or absent. So the way that the model is constructed is shown below in the graphical model. So we have here our latent ZS's. So the probability of a species being present in site S, we're going to represent by this psi S. We're also going to allow for some probability that that for the sites that are where the species is present, that we're going to record one of these confirmed presences. So it's not that we have confirmed presences for all sites, or that all sites that are where there is presence, we're going to get confirmed presence. So this is how we include ZS. But the main part of the model is further down. So we have these probabilities, theta 1s and theta 10s. So theta 1s is the true positive. So that's the probability given presence that we will observe, we will have eDNA in our sample. And then theta 10s is the corresponding false positive. So that's the probability that there'll be eDNA in our sample, given that we don't have presence there. And then we do the same thing for the lab stage. So given that we have eDNA in our sample, so that WSM is equal to 1, there's some probability that we'll record a positive PCR run. So that's P1, 1S. If we don't have eDNA in our sample, so if we have WSM equals 0, then we've got some probability P1, 0S of a false positive. And the structure of the model is that these probabilities of false positives at the lab stage, so P1, 1S and P1, 0S, those don't depend on presence or absence. They don't depend on ZS's or anything. They just depend on these WSMs. So we kind of separate out the presence at the site from eDNA presence from presence within the PCR or recorded presence within the PCR replica. So that's our basic kind of structure of model. So in terms of making inference in this kind of model, this is kind of complicated because we have likelihood symmetries. So what that means is we can swap around parts of the model and get exactly the same probability model. So there's various different solutions. So there's 4 different solutions. So the first solution is the kind of solution that I just explained to you, or the interpretation. But also what I can do is I can start swapping around parts of the model. So for example, because this model looks basically completely the same down any different kind of arm of the model, I can swap the θ1 1s with the θ1 0s and not really change anything. All I change is the interpretation. And so that's what these different solutions are. These different solutions arise because I'm changing the meaning from, so here at B, I'm interpreting it as a true positive. If I take 1 minus B, then that's going to be a false negative, right? So essentially what happens is through these likelihood symmetries, I'm giving different kind of meanings to the parameters of my model. So in a way, that's kind of not hugely problematic in some ways, because I can estimate the model and I can somehow come up with estimates, and then I could kind of rearrange them so that they look sensible to me. But as I'll kind of explain, in terms of doing that, which is a kind of pragmatic way of doing it, there's some kind of there are some kind of problems in a way. So that's kind of a quick fix. So since the model has these likelihood symmetries, it's only locally identifiable technically. And so the way that we're going to approach this problem is we're going to use Bayesian inference. And so our idea is that the prior distribution is going to give us the meaning to the model. So as I was explaining, the problem really is all these different solutions give different meanings to θ1 1, θ1 0, P1 1 and P1 0. And what we want to do is we want to impose the meaning that we think is the correct one. Okay, so it gives the right one of it picks the right one of these solutions always. Okay, and the way that we're going to do that is we're going to do it by placing a prior on pairs of parameters. So if we take θ1 1s and θ1 0, then θ1 1 is true positive for eDNA and θ1 0 is false positive. And so what we might think is that the probability of a true positive is larger than the probability of a false positive. Okay, and so that implies that we want to have a prior that encourages a large prior probability that θ1 1 is bigger than θ1 0. That gives a meaning to our model, that we imagine that true positives are more likely than false positives. And we don't consider that to be a strong assumption. So if that's not true in the data, of course, then the assumption is wrong. But as a kind of basic assumption, we're assuming that the test on the whole is not too bad. I mean, I think that's how I would interpret it. So technically, the way that we can do that is by using logit transforms. So we can take these parameters, take the logit transform, and then we can just define these normal priors. And by basically controlling the means of those normal priors, we can put prior information about the median value of θ11 and θ10. And then we can use this parameter, the variance. By changing the variance, we can control the probability that we get well, θ11 greater than θ10 or θ11 less than θ10. So this prior is parameterized by three things: A and B, which control the median, prior median of the true positive and false positive rate, and also the probability that the true positive rate is less than the false positive rate. And so we're going to choose this epsilon to be fairly small to encode that we believe that the true positive rate will be larger than the false positive rate. So if you do that, this is the kind of prior distributions that you get. So the black is θ10, so that's our probability of false positives. And so the median of that is 0.1. We can then change, well, we then fix this kind of discrepancy measure. So the probability of getting them the wrong way around, the prior probability of the error rates being kind of reversed, is small, so 2.5%. So you can change that to anything you want. And then for the other, we can choose the prior median for the true positive rate. And so here we've got a variety of different values, 0.8, 0.9, 0.95. And so you can see that we have this kind of, we get these shapes. So by changing the prior median of the true positive rate, you can see here with the red, we have a very large value. So this distribution is very pushed towards 1. For the blue curve, we have a lower prior median, and so it's more away from 1. And the black line is pushed towards 0. Okay, and so if the probability that the condition is reversed, so kind of the crossover part here between these curves is the part that's small. And so that's how we identify this model. So we don't formally identify it, but from a Bayesian point of view, we strongly favor one of these 4 solutions. Okay, so in a way, as I was saying, the practical way would be just to make inference and to kind of order them. So in a way, this is kind of a fancy Bayesian way of doing that. But really the usefulness, I think, of it is in terms of including covariates. So in covariates, then it becomes very complicated because we don't want to keep switching between these different solutions. And we want to be kind of sure when we look at the different variables, which solution we're getting. So we can extend this prior distribution into the situation where we have covariates by imagining that we're averaging. So once we have covariates, we're going to have we're going to effectively have different priors on different sites in a way. They're going to be highly correlated, but they're going to be slightly different. And so what we do is to average over all the sites. And so then we have the same kind of setup, that the probability of a false positive is a lot smaller than a true positive, but averaging over all sites. So that allows us to understand what factors are driving these error rates, and also to do variable selection. So we can do Bayesian variable selection and put that in as well. Okay, so there's some technical parts. So in terms of computation, we use, so we have a lot of logistic regression models effectively, and we can use what's called polygamma data augmentation to run an MCMC scheme. And so the advantage of doing that compared to other methods for logistic regression is that that avoids using trans-dimensional algorithms. So I'm not really going to go into why that's the case, but it avoids using these trans-dimensional algorithms directly, and so avoids using the reversible jump. and I think people who have used reversible jump often find that it can be quite complicated to implement because you have to tune particular proposals in these reversible jump methods. Okay, so Alex has implemented these methods in the RShiny app. So you can download the RShiny app from the web address here. Okay, so just to illustrate how the method works, we analyze some data on great crested newts. And for each of these samples, there were pond-specific environmental covariates, and we allow those to enter into the model at the two stages. So both the eDNA data collection stage and also at the lab stage. So we have one sample per site. So in our notation, that's M equals one sample, and we have 189 sites, and we had 12 qPCR runs for each site. Okay, so these are some results on the error rates. So of course, each site has different error rates because we have the regressors in there. But what we can do is that for most covariates, they were discrete, and so we could choose the modal values for those covariates. And so you can interpret these numbers as a kind of as the error rates for a representative site, some kind of average site in a sense. So we have psi is the probability of species presence, and then we have θ1, which is the true positive rate for eDNA, θ10, which is the false positive rate for eDNA, P11 is the true positive rate for the PCR, and P10 is the false positive rate for the PCR. And we're also able to construct 95% posterior credal intervals. So without this prior identifying the model, it'd be impossible for us to calculate these kind of credible intervals, really. We also looked at the effects of these estimates in terms of the number of samples that might be needed to detect species presence. So we have two sets of numbers here. So 1 minus psi x, that's the conditional probability of species absence given X positive qPCR replicates. And also we look at, so that tells us the probability of species presence given the number of replicates. And then we can also think about the conditional probability of having X positive qPCR replicates given species presence. And we can see how that varies with the value of X. So what we can find is that species presence is related to having a large number of positives, but also it's related to having a low number. And that's because we have these confirmed presences, then we know that there are some false positives, some false negatives effectively in our sample. So we have some sites where eDNA does not show up. Okay, so we're also able to do the variable selection. So we didn't find that there are any covariates that are linked to species presence or to the probabilities of errors at the data collection stage. We found that there were some related to the data analysis stage. So these are the things that we found that were important. So around presence of macrophytes and fish presence, and also some characteristics of the pond in terms of depth and the pond length. And so the pond depth and the presence of macrophytes have a positive effect on stage 2 true positive probability, while pond length and pond density have a negative effect. Okay, so as Doug and Eleni were saying, we're thinking about extending these methods into multiple species problems, metabarcoding. So that's metabarcoding. And so, okay, it's about metabarcoding, so I think we've been through that. So in terms of the modeling, we're interested in using joint species distribution models. So these are able to explain community composition whilst taking into account covariates and also spatial autocorrelation and so on. So statistically, these are multivariate logistic mixed models. And so some of the challenges really are around how we can model these false positive and true positive rates, and also to allow for potential of interactions between different species. So once we move to the multivariate case, things become a lot more complicated. And as we saw from the examples that Doug was looking at, then there's many species that we have to worry about. So statistically, we want to find an efficient way to represent the correlation between the species, and in order to do the inference. And so that's a very kind of challenging problem. So we're beginning to develop methods in that direction. Okay, so the work that I mostly talked about was this single species work, where we're interested in inferring site-specific probabilities of species presence, whilst also accounting for the probabilities of errors at different stages of the survey. And we believe that we've built a model that's able to overcome a lot of these identifiability issues introduced by the likelihood and to do efficient Bayesian variable selection. So one thing I should say around solving the identifiability problem is that in a way we don't really solve it, but we certainly alleviate it. So it's kind of it lurks there in the background, and being Bayesian, that maybe isn't a big deal because we have posterior uncertainty, and by using this prior we're able to interpret it. But of course, if we have other information, so the way that other people address this kind of identifiability problem is to introduce other types of side information, and that's definitely a valuable thing to do. So we've had these confirmed presences, and that was very useful in terms of getting a much better handle on the error rate. And also a thing that I haven't talked about here, but which kind of came up in the paper, is that we allow for these kind of replicate samples of sites. And we found that that really led to a much better kind of power to detect which variables are important or to detect specific effects. So, you know, in a way, having multiple samples at a particular site is really key to understanding these true positive and false positive error rates. So that's where I'm gonna end. So thank you for your attention.
Thank you very much, Jim. Obviously, I think the work is super interesting, having been involved with it. I also think you've explained it all very well, and so hopefully there'll be some questions from the participants on the model. So in the meantime, maybe I will just sort of briefly mention that today we've seen applications for single species, for multi-species, from looking at ancient DNA that Francesca looked at, or projecting things in the future like Doug did, and also Kerry talking about a much kind of a broader sort of application of DNA-based surveys for monitoring. And in all of these cases, observation error, so false positive and false negative observation error, are a challenge and a question. So I think hopefully people will kind of find this work interesting, and sort of start working with statisticians. And obviously the other issue that's come up a lot is the issue of study design, of how to allocate resources, how many sites, how many samples, how many PCRs, how many years, and so on. And so that's something that we've also been looking at, or Andy Baxton's been looking at now for single species cases. So I don't know if there are any other questions from people in the from the participants. So I don't see anything in the chat. I don't see any hands raised. Oh, there's something that's come up. So Chris, so he says he's got the same question as he had for Doug. I get that JSDM with imperfect decisions development. I'm more interested in how valid these results are. I'm not sure if Jim can understand what valid means, or whether you want further clarification on that, Chris.
Are the results valid? I mean, I think that's a slightly different question. I think the question we're trying to answer is what can we learn from the data, given that it's imperfect? So we're trying to kind of take into account the uncertainty in the estimation and the uncertainty in the measurement process. So I think then we have results that take that into account and kind of correctly, in some sense, adjust for that. So I think in that sense, they're valid. I mean, obviously, if you give me some terrible data, in a way, you're going to get terrible results. And in a way, I don't have a way of telling you that, other than just to kind of give it to you and say, look, these, there's just no information here, and my posterior looks like my prior. So that's kind of, I think, the level on which I would interpret it. It may be that people have more specific meanings of valid than I do.
I don't know. Let's see what. So Chris says, I mean, any inference from JSDM without a detection model. Totally believes the Great Crested Newt model. Yeah, so I guess that clarifies it. Yeah.
Yeah, I mean, I think I would say it's important to take into account, measurement error as much as possible, because otherwise, yeah, you have a huge potential for biases that you haven't control for. But I mean, in practice, people do statistics all the time, right? And they don't do that a lot of the time. So I think there's kind of on some level, as always with statistics, there's some judgment call about whether it's going to be important. And I think that depends a little bit on the processes that people use in the lab and to collect data, whether they kind of feel that they have very low error rates or not. But I think unless you have a very amazingly good process that's very robust, then you may well be, I think it's a good thing to take it into account. But I mean, obviously, it would be better for not to have any error rates, right? And to not have to do the statistics, actually. So better data is always better. but I mean, we can deal with also imperfect data. So there's a trade-off there.
Yeah, and I guess Naomi asked earlier on about their survey for Great Crested Newts. They've been doing it in exactly the same way, very standardized way, for 6 years, so that they don't have to worry too much about observation error. If they assume it's consistent every year, then they can still talk about relative occupancy rates rather than don't worry about absolute occupancy rates. But as she was explaining, technology has moved on since they first started doing this work. So should they now adapt the new technology, which they know is better in certain ways, but that would mean that their results, unless you build models now to account for the differences in observation error for the different technologies, their results will not be directly comparable unless you build models. So I guess that's
yeah,
something.
I mean, I think there's an investment in time in building models, right? I like building models, so to me it's a great idea. I can see for a lot of people it's a huge investment of time that they probably would much rather spend doing something else.
Yeah, absolutely. So Malcolm says, enjoyed the talk by Jim. I reckon that your Bayesian approach is potentially valuable also in the epidemiological context, also providing you have repeat sampling of individuals.
Yeah, no, I mean, I think there's lots of problems where you can apply this, and I think having repeated measurements, of course, helps you a lot, because it allows you to kind of get away from the prior, right? So that here the prior is, you have to accept that the prior is kind of informative, and really it's a price you have to pay of dealing with the data and not having the repeated measurements. So it's not a free lunch, yeah. And yeah, I mean, I think there's lots of areas where people could use these types of ideas.
Yeah, and so I think that's related to Richard's question about qPCR protocol for COVID-19, where, as far as we know, they collect a single swab from each individual and then they run a single qPCR, which, as we know, even with our informative priors, you can't overcome that identifiability. There's no replication at either stage.
No. And I think yeah, I mean I think that's an issue, right? So I think we saw a lot of that. So rather like the table I showed, that when you kind of do these Bayes formula type things and you look at this probability of you having the disease given a positive test, unless you've got a very low false positive rate, then you're in big trouble and you're going to just not detect anything. You're just going to have lots of people who don't have the disease and you'll say they are. So that's kind of a very first year statistics kind of thing that we see. But I mean that's really where it becomes a real thing, right? When people are trying to grapple with these kind of issues, policymakers, etc.
Yeah,
for them challenging.
I agree. I'd be really interested in getting some COVID-19 data, testing data to try things out. So if anyone has access or ideas of how to get the data. So Chibuso, sorry, again, I'm probably killing your name for the second time. Thanks a lot, Jim, for the excellent presentation. Did you control for the covariates, pond length and depth at both levels of the model? Sorry if I missed it.
Yeah, yes, we did.
Yeah. And then Nick asks, I wonder if you can use the same approach for looking at other survey data like camera trap data where images are auto-labeled and that will have a false labeling rate. That's very interesting.
I don't know too much about camera trap data, to be honest with you. Maybe Eleni is probably a better place to answer that question. But yeah, I mean, I think anywhere where you have false positives and false negatives and things like that, then I think that this is kind of a way to deal with that. The problem that comes, I mean, the problem just comes from modelling, right? So the natural model is unidentifiable, and you've got to do something to kind of fix it. And the Bayesian method is it's kind of a nice way to do it, actually.
Yeah, so I think if you remove one level, then you're just down to a standard occupancy model that relies on direct observation of the species, and you can then, as you say, Jim, just use the objective priors to informative priors to overcome the identifiability issue that's already been discussed by people who've looked at these from a classical perspective. So trying to find. So Laurie says, thanks for a great talk, Jim. I think we tried to ask some similar questions about great-crested newts using GLMMs in their paper published in 2019. It would be really cool to compare results.
Yep, sounds good. I mean, if we have an app, so we can, if you send us the data, or you can run the app on your data, whatever you feel happiest with, and then we can compare. That would be interesting, I think.
Yeah. So I hope I haven't missed anything. It's been quite a lot going on in the chat. Great. So Laurie says they'll keep in touch about the app. So I think I can see a few people now leaving kind of gradually. So I think we might just wrap this up. It's been an amazing meeting. So thank you so much, Jim and Doug and Francesca and Naomi and Kerry. I've gone backwards. Every single talk I thought was super interesting. They were all quite different, but obviously all related, but all really explored what different opportunities, especially looking at eDNA data and what we can learn about species. And there was obviously a particular interest, especially with community data, at least going forward. But I think we can agree that both single species and community level data are still important and will continue to be important because they each have their merit. And so it was definitely an underlying common theme to have proper models to account for observation error. Covariates have come in quite a lot. Both, for example, even we're looking at kind of current data, trying to understand populations and how they changed, how they look like right now, and also how to go in the future, as Doug was doing, or looking back, as Francesca was doing. So we're getting quite a few positive comments. Anything regarding the recording and so on of the meeting, then that's going to be dealt with directly from RSS. So I think the way it works is you'll get, once everything's been processed and ready, you'll get access to it. You'll get an email saying that this is all been processed and you can access it and watch it again. I think that's all from me. Again, thank you very much, everyone, and thank you everyone who took part in the chat. It was really very engaging discussions and conversations. If you want to, I'm sure the speakers would be more than happy for you to contact them with more questions. And if you can't find how to contact them, you can always contact me and I'll contact the speakers on your behalf and ask for their permission to share their contact details with everyone. And I think that's all. Thank you very much. I really enjoyed it, and I can see a lot of people enjoyed it as well. And it was a virtual meeting, so we didn't really have the chance to maybe network as much as we could during the breaks, but hopefully we're all used to this now, and if there's anything that you learned or you want to explore further or you want to collaborate on with people, then I'm sure we can work out ways to do it. And Luz's emailing, chatting about letting her know if you don't want to be included in the recordings that will be circulated, then let us know, let her know. And other than that, thank you very much, everyone. I think that's the end of this meeting. Bye.